Hello out there, all you YouTubers. This is Jonathan Richwine coming to you live from Rocky Top, Tennessee in Knoxville. Today I'm going to be speaking with you on grazing native warm season grasses and focusing on part of my PhD research here at the university. I'll also be talking about how to actually analyze some of the statistics in our studio. Why are native warm season grasses significant for the Mid-South? Well, cool season grasses dominate the landscape around here. And when I'm talking about cool season grasses, I'm primarily focused on tall fescue. Now, cool season grasses in and of themselves are more nutritious than warm season grasses. However, they're not more nutritious when they're trying to be grazed during the summer months while those cool season grasses are dormant. So by incorporating a warm season grass into a cool season grass mix, you're able to get an overall greater forage quality throughout that pasture, which in turn is definitely going to turn into dollars for that producer. There are multiple advantages to using native warm season grasses. They have greater water use efficiency, greater nitrogen use efficiency, they're deep rooted, and they can handle the stressors of our environment here in the South. Primarily the drought tolerance. If you look at the picture on the right, it shows 4 native warm season grass species of Indian grass, big blue stem, switchgrass, and little blue stem. And it shows a turfgrass exotic species, which for this example we will call tall fescue. The native warm season grass roots go for all the way from 0 down to a depth of a little over 10 foot according to that picture. And the turfgrass exotic of tall fescue does not even go a foot deep into the soil. So those native warm season grasses are able to mine for those deep nutrients and those deep water pockets. There's also an environmental benefit to using natives. They provide food and habitat for various wildlife species, such as the northern bobwhite quail, the eastern cottontail rabbit, pollinators such as bees and butterflies, as well as cattle, which is primarily what I'll be focused on. But we also want to look at the soil health aspect of these grasses, because they do provide places underneath the soil for soil microbes to live, and fungal hyphae actually live inside the roots. For the first part of my research, I'll be focused on native warm season grass pastures as affected by rest from grazing pressure of cattle. So the objectives of the study are to evaluate whether rest periods from grazing affect forage nutrient value, have an effect on the establishment, persistence, flowering, and seed set of various forbs and legumes, and if they affect the actual soil organic matter and the soil macro and micronutrients. Diving into the materials and methods, this study will actually take part at the Research and Education Center in Greenville, Tennessee between 2018 and 2020. So the test has actually not gone in yet. But we'll have 2 experiments there, and I say 2 because they're just mirror images of one another, but the pastures are a native warm season grass switchgrass pasture, and then the other experiment, which is another pasture of a big blue stem Indian grass mix on a 1-to-1 ratio. And both of these pastures will be planted with a biodiversity polyculture of 11 warm season forbs and legumes. Each one of these 2 pastures are roughly 3 acres in size, or 1.2 hectares if you enjoy the metric system. Now the study design is actually a completely randomized design with 4 replications. The 5 grazing pressure treatments within inside each pasture are no rest, early rest, middle rest, late rest, and an actual no grazing control. And the rest intervals are put in there so that we can actually identify whether the forbs and legumes are able to develop and potentially flower and set seed to benefit those pollinators, as well as to assess the regrowth and biomass quality of the native warm season grasses. So here's a design layout. On the left you have the switchgrass pasture, and on the right you have the big blue stem and Indian grass pasture, with all 5 treatments being in there randomly. So to break down the actual treatments for you, there will be a 12-week grazing period. starting roughly around May 15th and going to August 7th, which will give us 84 days of grazing. So the no rest treatment means that cattle are able to graze those paddocks for the entire 84 days of the experiment. Early rest means the first 28 days is blocked off to where cattle cannot graze that, but can graze that paddock starting the 29th day and going to the end of the study. The middle rest is as follows. The middle days of the 29 day through 56 days, the cattle cannot graze those, but can graze any days prior to and any days following. In the late rest, they can graze up to day 56, and then on that 57th day we're going to close them off to where they cannot get on those paddocks at all. And the no graze means those paddocks will not be grazed at all throughout this 84-day grazing period. So to give you a better example, this is just of the switchgrass pasture. The light green shows the area that the cattle are allowed to get into, whereas the dark green or brown shows the area that is blocked off from cattle being able to get to at any specific period. The early being on the far left with the 1 to 28 days, the middle being in the center here with the 29 to 56 days, and the late being on the right with the 57 to 84 days. So in grazing these pastures, and specifically in the switchgrass, we will have 5 6-weight steers as our testers, and we'll be putting them and taking them off of there based on a forage availability. So as the grass grows throughout the summer, it might get more than our 5 cattle can handle, so we'll have to add on another one to make it 6 and 7 and so on. Or at the end of the season, as the grass starts to go dormant, we might have to take cattle off so that they're not putting too much pressure on there, because we want to keep the canopy height for the switchgrass in between 15 and 18 inches. The same format is used for the big blue stem and Indian grass pastures. We'll start with 3 6-weight steers as our testers, and we want to keep that canopy height between 12 and 15 inches. The data we plan on collecting from these pastures, we will collect prior to and at the conclusion of grazing. So we will take plant counts, not only for the total number of plants, but for the species. Is there 1, 2, 3 through 11 species within the side of that square? And we will take those on a 1/4 meter squared basis at 5 random locations within each treatment. We'll also collect data from forage samples using near-infrared reflectance spectroscopy to actually get the forage nutritive value parameters of acid detergent fiber, neutral detergent fiber, crude protein. We want to know the actual number of head grazing each one of those pastures throughout the entire experiment. We'll take visual ratings prior to and following each rest period on a 1 to 5 scale to actually see what the weed pressure is in there. And also soil samples will be taken prior to the initiation of the study in 2018 and at the conclusion of the study in 2020 at 3 levels, starting at 0 to 5 centimeters. Then the next level is 6 to 15 centimeters, whereas the third level is 16 to 30 centimeters deep. And we want to know the soil carbon, the cation exchange capacity, and the macro and micronutrients within each one of those levels. Some of the statistical analysis will be the response variables of forage nutritive value, soil organic matter, cation exchange capacity, soil macro and micronutrients, and those plant counts. And we will run those on a ANOVA test, and they will be considered significant at an alpha of 0.05. And we'll either run a pairwise comparison of Tukey's honestly significant difference or a Bonferroni correction. And the fixed effect will be rest periods, with the random effect being years. Now, since we have yet to conduct this study, I have created a mock example of the data that we could collect from this. So here we have our 5 plant counts, our 5 counts at the end of the actual year. And this is going to be how many species are within each 1/4 meter squared. And here in the middle we have our treatments, 1 to 5, with 1 being no rest, 2 being early rest, 3 being middle rest, 4 being late rest, 5 being no graze. And we want to see if any of this is significant. So let us use RStudio, and we'll actually title our data up here as a biodiversity. Excuse me. Sometimes my mind works faster than my fingers. Grazing experiment. Biodiversity grazing. So, in order for us to use the data that we have created in our Excel spreadsheet, we're going to copy it, go over here to R, and we want to type in, I'm going to call our data, when you need a data name, so I'm going to call it graze, with an arrow, and then we want it to read that data, but now we need to tell it where to read it from, and since I copied it, I'm going to have it being read out of our clipboard. And there is a header on that data, so I'm going to make that true, and that just shows that, as you can see at the top of each one of the columns, I have a heading there. So if you put header as true, that means that that data, that the program will not read the heading labels as the first part of your data. And so in order for R to read any one of your lines you put into the input statements, you have to hit Control Enter. And as you can see at the bottom here, it came up in blue, so that means that it read it as a good statement. And over here on the right, it actually shows your data has 20 observations of 3 variables, which our 3 variables were plot, treatment, and species. So all that's good. But to actually see what we did, let's type in our data name and hit Control Enter again, and voila, it actually came up correctly. So far we're cooking with grease. So now to actually see if it reads our data the way we want it to, we want to see the actual structure and see what it has labeled our variables as. So let's type str for structure, and then we have our data labeled graze. So now we hit Control Enter, and at the bottom here in our console it shows that plots are integers, which are fine because we don't necessarily care about that one. But it also shows our treatment is an integer, which our treatment is actually a factor, so we'd have to change that. And our species is read as a number, which is good because it's our response variable, so that's what we want it to be read as. So going up here to actually change the treatment to a factor, you got to type in your data name. And we want to locate our inside our data, so give us the dollar symbol. Go down to treatment, and we want it to be read, got to get that arrow, want it to be read as a factor, as dot factor. And that was in graze treatment. So now let's hit Control Enter. Says that it is a factor in our console, but let's go up here and see our structure one more time. And it did work. Integer, factor, and number. Those are exactly what our parameters need to be read as. So now to actually just see our data, let's make a box plot, a simple box plot. Then we need to have a model statement within here, and you want your response variable first, which for us is species. And then you have a tilde, not an equal statement, but a tilde. Then you want the actual independent variable of treatment. Where's it going to find this data? Well, it needs to look in our data statement known as graze. And I'm going to go ahead and label it, label our axes, so that way we know what they are showing us. So our y-axis is number of species, and that was on a quarter meter basis. And our x-axis, so our x label, is treatment. Okay, so now let's see if that pops us up a box plot, and it does. That's a pretty box plot there. And as you move the center over to the left or to the right, it actually makes your box plot larger or smaller. Now if you have it over too far and you try to get this to read, it actually worked now, but sometimes it won't read it. And then you will see an error statement, such as at the bottom it just came up. And that's just strictly because this area is too small for it to put a box plot in. So move over your middle, because it had me freak out one time before, and do it again, and it shows up. So to actually see if these are statistically different, you don't need a box plot. You need to actually run an ANOVA table. So we want our model to be of a linear fashion, linear model, with our model statement again of species, treatment, and that is in data grays. Control enter to make sure it read that. Now we want to summary of an ANOVA, and we want our model to be read for mod, so control enter, and down in our console it shows that we have a significant difference, as you would expect from this box plot to your right, but now the ANOVA actually tells us. So to actually see that significant difference broken out, let's run a pairwise comparison, pairwise t-test. There we are, of our treatment level, but you got to show it the response variable that it needs to read, which is species, and we want it to actually work for our treatment. And for Dr. Zobel, we are going to adjust it using Bonferroni. Show us the actual breakout. And down at the bottom of the console, I will make it go up a little bit for you. 1 is significantly different from 2, which is 1 is actually significantly different from all of them, with 2, 3, and 4 not being significantly different from one another, and 5 is also significantly different from all the rest. So we have a breakout there where the 3 are in the middle that are not significantly different from one another, which we would expect from this box plot, as well as 1 and 5 are significantly different from all the rest. So that is how simple an ANOVA table can be using R. My advice to you is to not let it get over your head, and if you need any help, go to Google. With that, I would like to say thank you for all your listening and for listening to me rant. Go Vols.